Hi, everybody. Thanks for joining us today for another interesting talk about PFAS. So today we're going to be talking about PFAS in people. So our speaker today is going to be Craig Butt. He is the Applied Markets Manager for the Global Technical Marketing Division in SCIEX. So Craig has over 20 years of mass spec experience. He received his PhD from University of Toronto, where he investigated PFAS in biological systems. So very similar to what we're going to be talking about today. After completing his PhD, he went to Duke University, where he was a postdoctoral researcher and a research scientist, where he investigated the phosphate-based flame retardant exposure in children and adults, along with measuring PFAS in human serum. So obviously very relevant to what we're going to be going over today. So with that, Craig, if you're all set, feel free to start.
Sounds good. All right, thanks, Carl. As Carl mentioned, today we're going to be talking about measuring PFAS in humans, in human tissues. So specifically looking at serum. And again, this is part 4 of our 5-part series. I encourage everybody to go back and look at those previous sessions and then join us for the next one in just a few weeks. So just a kind of a rough run through of what I'll be talking about today. I'll do a little bit of a PFAS introduction. I'm assuming most people are familiar with PFAS, so this won't be an extensive introduction, but I'll really focus in on water as well as serum analysis and how those all kind of link together in terms of the PFAS life cycle. We'll be starting off talking about measuring PFAS using nominal mass quantification. So on triple quad or Q-trap type of instruments. And then we'll be talking about PFAS being measured with the QTOF or the accurate mass instruments. And the particular advantages that accurate mass can provide you for this type of PFAS and serum analysis. So specifically through elimination of biological interferences, and those that do this work are familiar with that, as well as reducing the matrix background. And then kind of the final step of the presentation will be looking at how non-target analysis can be used for PFAS serum measurements, specifically using a fragmentation pattern to increase our confidence in the ID, and then as well as kind of an interesting surprise when we looked at some blood samples finding a so-called unknown PFAS compound. All right, so of course, this work was possible through the help and really work done by a lot of other folks. For the nominal mass work that I'm going to be presenting, both Pierre and Scott here at SCIEX produced that work. I'm just presenting it on their behalf, frankly. And then for the accurate mass work that was done in collaboration with our partners at NMS Labs, Fred and Joe, and then as well as Casey Burrows from SCIEX. So again, all of this work that I'm presenting, wouldn't have been possible without their contributions. All right, so looking at a little bit of PFAS introduction, I always like to start with this slide because it kind of takes us back to the fact that PFAS were manufactured really since the 1950s, but our awareness of the problem didn't start till just over 20 years ago. And really these two groundbreaking studies from 𝗔𝗱𝗀𝗀𝗀 and then Hansen et al. Showing that PFAS could be measured both in wildlife and in human serum. This one started out human serum for the United States, but certainly we know we can measure PFAS in human serum from throughout the globe. And this showed us that PFAS were kind of getting into the environment, building up through the food chains, and then accumulating quite high up. But this really wasn't the first indication that PFAS may be in human blood. And that was work that Taves did back in 1968, reporting evidence for organic fluorine compounds. However, at that time, the analytical technology didn't exist to actually identify what they were. We now suspect that actually what he was measuring was PFAS, but we didn't know that at the time. And so, a lot of attention has been put into measuring PFAS in drinking water. Really the reason for that is that drinking water can be a significant source for PFAS contamination and accumulation in humans. I just put up this study from 2016 showing the widespread contamination of drinking water supplies in the United States, but certainly our knowledge of this kind of contamination has grown, and this is clearly not just isolated to the United States, but globally. In terms of regulations, there's various drinking water guidelines, and then probably some regulations to come down the pipeline in the future, as well as not just federally, but state-specific or region-specific guidelines or regulations. So again, we spent a lot of time talking about drinking water, and then today we're going to be talking about human tissue, specifically blood. But of course, there is a link, right? And it is important for us when considering PFAS to look at the entire PFAS life cycle. I've shown just a couple of good cartoons here, schematics, to show how all of those different environmental compartments are related and why it is important to look at those when we do consider human blood or other human tissues, such as breast milk or cord milk or even urine. So all of these kind of link in together in kind of a very complicated way, and we won't spend our time diving into it. But ultimately, with the interest leading to human blood or human tissues because of a potential toxicological effect. I thought this study was really interesting here, from Califat at the CDC. This is looking at the US NHANES data from 2013, 2014. And what you see here is the percent detection, right, on the y-axis of various PFAS compounds here on the x-axis, and looking at here in kind of the lighter blue for serum, and then the orange or rust color for urine. And what you can see here, well, first of all, for serum, certainly some of these PFAS are detected in nearly all of the samples. You see here nearly 100% of the samples. Whereas with urine, right, very, very few detections, right? So showing us that serum is really a superior tissue for biomonitoring of PFAS exposure. So that's kind of the first thing that I gleaned from that. The second thing that I gleaned from that is the fact that you can see that some of the compounds, right, there's sort of a sweet spot here where, some of these, let's say, moderately chain length compounds are the ones that are accumulating and we're detecting, whereas, very short ones or very long compounds are not as accumulating as much, right? So this, of course, gets back into bioaccumulation potential, but there certainly is a kind of a sweet spot for compounds that are most predominant in human tissues. And again, serum tends to be the one which the PFAS will accumulate in and is in a sort of a preferred tissue for biomonitoring above something like urine. All right, so that was more on the nominal mass side, but of course we can use accurate mass techniques to try to understand what else may be in there, as well as using this fragmentation pattern and looking at those patterns to confirm the identity of what is being detected. And here's kind of, this is another example, work out of Sweden. Again, showing here kind of a cyclic PFAS compound and using the fragmentation pattern, the MSMS fragmentation pattern, to in fact confirm its identity. This is a really interesting graph here, and I'll just take a minute to explain this and why I think it's important. So this EOF, or extractable organic fluorine, is kind of a measure for the total PFAS that are in the serum. What the graph is actually showing is the percent of compounds that we can identify as compared to the total potential that's in there. And what you can see is that we're actually declining with time. And so what it's saying is that we're doing actually a worse and worse job at identifying which specific PFAS may be in human blood. So what that is to say, and in my opinion, is that there is a large potential or a large reservoir of unknown PFAS that are in human tissues that we don't know, right? We don't know for sure is there. And I would make the argument that if we are interested in fully protecting human health, then we need to understand specifically what's in there. And that's a role that some of the accurate mass techniques can help us with and help us understand what's there. Okay. The other thing too is, the landscape of PFAS is constantly changing as our awareness of what is being used in products improves. So does our awareness of what's in the environment, and therefore what humans are exposed to is improving. And here's a study from down in North Carolina about some of these novel ether carboxylic acids that are being detected down in that area. And then what is interesting is that when measuring the PFAS in the blood from the residents around Wilmington, North Carolina, that almost a quarter of those are of these novel compounds, right, that we're just starting to understand over the last few years. So this, again, I think, makes the strong argument that our monitoring lists need to continue to evolve with time as our understanding of what humans and indeed wildlife are being exposed to over time. Okay, so just kind of a summary as I see it, and specifically related to serum analysis. Really it was suspected that back in the late 60s that PFAS were in our tissues. But we didn't have the specific analytical techniques to really confirm and even measure what's there until really the early 2000s. And then those early monitoring lists and the early studies may have focused on just some of the kind of smaller compounds, but again, as the landscape of PFAS is constantly changing, right? And we need to start looking at some of those more novel and replacement compounds, right? And then I would also then finally say that there is a good argument for using these accurate mass techniques to try to understand the unknown or unidentified PFAS that seems to be in blood. All right, so now the first technique that we'll talk about is the targeted serum analysis. So this would be quantification using triple quad or QTRAP type systems. So a question we often get is on sample prep, right? You've got a serum sample or even plasma sample. What should we do to extract it and get those PFAS out? There is a lot of techniques, I would say, that are published in the peer review literature. A lot of really good methods that are out there. Not really one that I would recommend, let's say, above another. Part of it depends on the sort of detection limits that you're trying to get, as well as the volume of serum that you may have accessible. Some of the techniques that are there are your classical kind of protein crash with a methanol or acetonitrile, as well as using something like an SPE concentration step or cleanup step. So lots of techniques that we can use. Now, modern technology, certainly since I started doing this, back in the mid-2000s, our advancements in technology has allowed us to achieve much lower detection limits. As well, it has allowed us to use smaller volumes of serum, right, which is often important in terms of its practical handling as well as sampling possibilities. But again, as well, one thing with using advanced and sensitive technology is the ability to dilute out sample matrix effects, right? By getting that enhanced sensitivity to allow you to dilute out what may be still left over in that extract. Okay, so I'm just going to run through some methods here for a technical note from Pierre and Scott at SCIEX. Really good work here to look at PFAS using, in this case, the 6500 plus QTOF. In this case, 22 PFAS compounds were measured during the analysis. A very small amount of serum, you can see here, 50 microliters was used, and I'll show you what kind of detection limits they were able to achieve with that. And the sample prep, I'm going to go into details on the next slide, but involved a protein precipitation with acetonitrile. And so here are the methods. I don't want to kind of get into too much of it here, but really what I want you to glean from this is that it's a fairly simple method in terms of the preparation, kind of a protein crash, and then making up in buffer. So fairly straightforward. Again, using only 50 microliters of serum. This is something that is really common to all PFAS analysis. And most people are familiar with the concept of using a delay column, also known as a holdup column. Particularly when you are trying to achieve those really, really low levels, low detection limits. It's important to control first of all, the PFAS that may be leaching from the LC system by removing as much of the fluoropolymer that may be there. But then there's always going to be a little bit that's leaching out from things that we can't replace. And then using this delay column to delay that pump contamination from the analytical peak is essential. And you can see here just an example. This big hump here was what was delayed from the pumps of the LC system. And if we didn't do that, then that would have resulted in much higher detection limits. So again, not just for drinking water analysis, but any PFAS analysis, it's important to control the contamination from your LC system. Okay, and this is actually kind of another example showing things a little bit more detail here with the heptanoic acid and the hexane sulfonic acid. And you can see here this in the yellow, right? And so that would be what is being seen in the delay column. And what if we didn't have the delay column? You can see here in the red. And so, if we didn't have the delay column, that would be the background contamination that would effectively raise your detection limits. But with the delay column, you can see that blank contamination is now gone, right? It's held up. And then here you can see nicely a 10 ppb standard compared to the delay. So again, just sort of a little bit more detail in terms of the chromatograms. Without the delay column, often you will see an increase in apparent blanks coming from the LC system. Okay, so looking at some of the levels that this method was able to achieve. Now, I will stress kind of going forward, What we've got here is, in fact, these are spiked samples. So these are levels that are not in the vial, but kind of representative of what's in the serum. So you can see here our matrix blank for the hexane sulfonic acid, nice and clean. With spiked, we are able to see down to 0.1 nanogram per mil or 0.1 ppb, right? So quite low levels there. If we look a little bit more detail, in this case for PFOA, for both the quantifier and qualifying MRM, again, nice and clean with the matrix blank, and then able to see down to, in this case, 0.5 nanogram per mil. In most cases, the lower limit of quantification was in fact 0.5 nanogram per mil or ppb, and only I guess 1 or 2 analytes slightly higher. So again, using 50 microliters only of serum on the 6500 plus QTRAP system, we're able to get quite low detection limits in the human serum and clean matrix and clean backgrounds. Okay. So now what we're going to do is spend kind of the rest of the time looking at accurate mass techniques for serum analysis. And, for those that are not familiar with an instrument such as a QTOF, this was work that was done on the X500R QTOF. And you can just see here kind of a picture of what it looks like in the schematic. The key really for this is the front end source is the same that's on our mid-range triple quads, which means we're getting a very robust source, able to tolerate the complex matrix of a serum, but also high sensitivity. As well for those that don't have a lot of space in the lab, this is a true benchtop system, right? And so this will go on your benchtop, not really much bigger than a standard triple quad. And so, the difference of a QTOF versus a triple quad is, for that third quadrupole, it's replaced by this time of flight chamber rather than a quadrupole, which means it has very fast scanning capabilities. And as well as we can know the mass with very high accuracy and very high precision, down to kind of the third or fourth decimal place, based on its mass resolution. So that provides a lot of advantages in terms of interference removal, as well as confirmation. And I'll show you that in the coming slides. Okay, so just to kind of cover that again too, for those that may be new to accurate mass instruments, the front end of the system is identical to a triple quad in the sense that there is a Q1 which will filter the precursors, and then there is a collision cell where you have that collision-induced dissociation to break apart the precursor. And then instead of filtering out for a single fragment, we're able to use that time of flight chamber to scan out all of the fragments, but at very high precision and very high accuracy. So what it means is that you can know the precursor mass very accurately and precisely, as well as all of the fragments that are formed. And so for us doing that kind of work, what I'll be talking about is using this non-target acquisition for suspect screening and using the fragmentation pattern to really confirm what we're seeing, as well as doing some unknown ID. All right, so the first thing that I want to cover is looking at how we can use accurate mass techniques to resolve some biological interferences. What do I mean by that? This has been known for actually quite some time, right? Since that original paper from Hansen in 2001. And we know that some of the perfluorinated sulfonic acids, such as PFOS or the hexane sulfonic acid, With that 80 fragment, which is the SO3 fragment, we know that there is biological interferences. And I've kind of listed those down here as a 𝗏 acid or the other pregnenolone sulfate isomers. And, that's been known for quite some time. And some additional work from John Benskin in the later 2000s and now others that have followed up on it, did do a characterization to try to understand what those were. So typically what we would do is instead of monitoring the 80 fragment, right, the SO3 fragment, we would monitor the 99 fragment, which is the FSO3 fragment. However, though, what we can also do is we can also use the accurate mass instrument to mass resolve those interferences away. And I'll show you that, how we can do it. Okay, so let's look at PFOS to start with and look at those interferences. Okay, so to orient you on this figure. So this is a chromatogram of the 499 precursor, right, for PFOS. But the extraction width is 0.8 Daltons, which is representative of what your triple quad would see. And what's interesting is that we see the characteristic branched isomer and linear isomer peaks for PFOS. But what we also see actually is that there is other interferences that are in the chromatogram, these other peaks here. And so in fact, when we look at the masses of compounds that are under these PFOS peaks, right, we can see that there are some other masses here. So this one here, this 498.9300, that is the exact mass for PFOS. And you can see here there are some other compounds that are present. In our case, we are able to use a chromatographic gradient that's able to mostly separate those out, but not fully. And what I want you to pay attention to is, hopefully you can see it, is that these interferences are really different from our PFOS in the decimal places, right? And so if we were only looking at it from a nominal mass perspective, right, to 0.8 mass resolution or even 1, then all of these interferences would be seen. But because we have a high resolution accurate mass instrument, we can actually mass resolve out those. And then if we only look at the specific mass of PFOS, right, we would obtain a clean spectrum. Okay, so how do we know that some of these other peaks here are not just other PFOS isomers, but in fact are some biological interferences? So remember, these two are the PFOS, the branched and linear, whereas if we look at the mass spectrum under this peak here at 3.5 minutes, we can see that our PFOS mass peak is not there, right? It's really, this is at something at 498.29 or 289. And when we look at the formula associated with that is consistent with this 𝗏 acid compounds. And in fact, if we go and we do further work and we look at the fragmentation pattern of those interferences, the fragmentation pattern here shows that they are consistent with what we know of in terms of known MSMS database entries for 𝗏 acid. So again, this demonstrates that we can use the high resolution instrument to mass resolve those interferences. And in fact, we know what those interferences likely are. Okay, and so again, the initial chromatogram I showed you with all the interference peaks there, that was kind of representative of what a nominal mass instrument would look like or would see. But here, if we use the ability to very accurately and precisely pull out the masses. So in this case, using a resolution of 0.02 or 20 millidaltons, you can see we're only pulling out PFOS and we're not also pulling out those potential biological interferences. So that's with PFOS. We can do the same thing if we look at the hexane sulfonic acid. And here again, it's the same kind of diagram as I showed last time. If we look at the hexane sulfonic acid, but use a wide extraction width, you can see there's multiple other peaks that are in there. Here again, this is the hexane sulfonic acid at the exact mass, but here there are some other interferences that are present under the peak. They're not as large because we were able to use a good chromatographic gradient, but they certainly are present in our nearby. So then if we look at one of the other nearby peaks, you can see that it's certainly not hexane sulfonic acid, but indication of this compound here, which Benskin identified in 2007. So when we look at it and we try to understand a little bit more detail what may be there, it's kind of hard to see or hard to identify. We don't have a consistent match in a library database, so we don't know exactly what it is, but we do have some indications as to what the structure may be based on the fragmentation pattern. And again, if we take those, that accurate mass, extract it with that 20 millidalton window, you can see those interferences are not present. And that's taking advantage of the high resolution instrument. All right. So, so that's one technique for using accurate mass, really is to, mass resolve, get rid of those biological interferences, which ultimately may cause false positives, and take advantage of that. You'll notice that I was able to use a certain chromatographic gradient to try to separate them out. That may not always work, right? And so, perhaps we were fortunate that we were able to do it with this specific case in this sample, but it may not always be possible. So that's the advantage of using a high resolution accurate mass instrument to remove those interferences. So now we're going to talk about something a little bit different. We're going to do non-target acquisition, and we're going to use a fragmentation pattern, much like you would with a puzzle, to confirm the identification of what we're actually detecting. So in this case, again, I won't bother to dive into all of the details with how this works, but we use a technique called SWATH, data independent acquisition, and this is used to generate the MSMS spectra. So essentially what it's doing is it's taking all of the precursors in small little windows, and it's fragmenting them with your collision-induced association, getting that fragmentation pattern, and then performing some library database searching to confirm its identity. And this SWATH and in using a variable SWATH window acquisition is unique to SCIEX, and allows us to get really clean MSMS spectra, reduce the complexity for that database searching. All right, so what kind of data do you get, and why is this advantageous over using just a standard triple quad technique? So running it on your triple quad, right? If you had a standard, I always like to think of multiple lines of evidence to confirm what's in a sample, right? And so generally what you would have on a triple quad is you would have a retention time. If you were fortunate, you would have both quantifying and qualifying ions, and so you can do a ratio of those. So you would have an ion ratio and a retention time, right? So 2 lines of evidence. However, with an accurate mass instrument, we also have the retention time. We have the precursor or fragment mass error that we, again, I'll remind you, we can know very, very accurately to the 3rd or 4th decimal place. So that's line of evidence number 2. We can look at the isotope pattern of that precursor, right? So number 3. And then again, if we have the fragmentation pattern, you can see here for PFBS, the butane sulfonic acid, we can take the blue, which is our experimental pattern, compare it against a known database, obtained from a known standard, and we can see that all of those fragments match up perfectly. And so we have a 4th line of evidence. So again, when you're thinking about increasing your confidence in terms of identification, multiple lines of evidence are key. Where do those library databases come from? SCIEX has a really comprehensive library for PFAS, over 250 compounds, covering a wide range of PFAS, including a lot that come from the 𝗔𝗱𝗃𝗃, the firefighting foam mixtures. And so that's what we use as our database. Here is for our serum samples. These are actually pooled serum plasma samples. And you can see here, we found something, right? You can see a nice big peak. The mass of it was consistent with PFBA, the butane carboxylic acid, right? And you can see a really, really strong mass error. The isotope pattern was good, but most significantly here, right, we get a good library entry, or good library match, right? So we're very confident that we're seeing the PFBA. This here, another example to show you for the C9 carboxylic acid, right, which are, the PFNA. We've got, again, a nice strong peak. In this case, a really good mass error, the isotope pattern, and then the MS-MS match here. So even at a low intensity, we're getting a good match of the fragmentation pattern. Okay, all right, and so another one, this is actually not one that would be on a lot of monitoring lists, right? We often monitor the hexane sulfonic acid, which is the C6, and the C8 sulfonic acid PFOS. In this case, this is the detection of the C7, right? Heptane sulfonic acid. And much like other compounds originating from electrochemical fluorination, we do have a branched isomer peak as well as the main linear peak. And again, we've got a good mass error and really good fragmentation pattern match. Again, confirming the identity of what's in our sample. All right, so here's one which was a really interesting case. So this is a PFOS replacement chemical that was on our screening list. You can see the name up here, right? The 9CL compound. Those doing the EPA monitoring for drinking water are probably familiar with this. And we threw it into our screening compound and our screening list, and you can see we got a decent hit. It's not very intense, but there is definitely a peak there. We have a very good mass error, so we suspect it's there. What's interesting about this compound is that it does have a chlorine, and you can see from the isotope pattern it was suggestive of a chlorine in that molecule. But you'll notice we didn't have a match in our library. And the reason is that this is a fairly new compound for most people, and it's just not in our database. So we thought, well, what else can we do to take that extra step to confirm what's in the database? And we did that by running a standard and performing that fragmentation so that we had it in our now database that we could then search against to confirm. And lo and behold, right, when we have that MS-MS in our database, that we do have a good library match, right? And so we are pretty confident now that in this blood sample, be it at lower concentrations, that this 9Cl compound, this PFOS replacement compound, is there. So definitely something that folks should start looking for if they're not already in their human samples. Okay, so just to kind of summarize what I showed today and just kind of wrapping up everything. So hopefully I demonstrated to you that both nominal mass, triple quad or QTRAP instruments, as well as the accurate mass instruments, can both be used to monitor PFAS in serum. Certainly there's sort of advantages to both kinds of instrumentation, and hopefully I was able to distinguish between the two. The nominal mass instruments, very sensitive techniques, allows you to get those sub-nanogram per milliliter or sub-ppb levels with only using a small volume. Some of the advantages of the QTOF, the QTOFs are still quite sensitive as well, but the added advantage of mass resolving certain biological interferences in the serum. And then hopefully as well, I've convinced you on the utility of using a fragmentation pattern, an MSMS fragmentation pattern, to increase the confidence in what you're seeing and what you're identifying, which is particularly important when you're looking at some newer compounds in which we don't have as much knowledge about. And then also too, finally, what I will do is stress to you to come back in 3 weeks, right, and listen to the final part 5 of our PFAS series. We'll be looking at different matrices, such as environmental matrices, food, as well as consumer goods, such as textiles and cosmetics. So some really interesting stories there, and help us understand what else is in the environment and the PFAS life cycle. So yeah, please come back in 3 weeks. And with that, thank you so much for your time and attention.